Okay, so this will be the third hour of the lecture, which involve macromolecule. And your job to study for the exam next week is to think of what are the four macromolecules and what are the component that makes of these macromolecule. And you can think of these macromolecules or polymers, right? That mean poly mean many, mer mean unit, right? So what are the monomers then make these polymer, right? So you can think of the polymer. If you break down the polymer, you get the monomer, right? Okay. So that's it in that context. And of course, do you guys know what the four macromolecules are yet? What are the four macromolecules?
Protein, lipid, carbohydrate, and nucleic acid.
Very nice. So all of those four macromolecules always have carbon, hydrogens, and oxygens to them, right? And some of them, particular protein, has nitrogen and phosphorus in them. Um, but they do have some functional group, right? Some of the functional group, I give you an example, carboxyl group, right? That's an acid, right? So in other words, a carboxyl group looks like this. That's a carboxyl group. Well, think of carboxyl group as an acid, right? And again, an acid is any molecule that give off the hydrogen ion. It has an amine group, which is this. Okay, that's an amine group. So why do you call it amino acid, for example? Because it has one N has an acid like this, and here's the amine group. I'm just going to switch this to say, oh, NH2 here. Because carbon has four unpairs electron, it can form four covalent bond. That's a hydrogen. And this right here, I consider is R, right? In other words, this is an amino acid. This is an example of amino acid. Why? Here's the amine group, here's the acid. And in our body, we have 20 different kinds of amino acid to make the protein. And how does they differ from each other? Is this position here called R. The rest of them stays the same: acid, amine, hydrogen, and the R. If it is a CH3, for example, a methane group attached to that, that is known as an alanine. If it has a CH, CH, and its carboxyl group to it, uh, that is considered as glutamine, um, glutamate, glutamate. Now, the point here is that keep in mind that this side chains is critical. Right? This right here is critical. Some of the side chains is polar, and some of them is non-polar, which I'm going to show you a little bit. And that's how amino acid might be differ from each other is because of this hydrogen, the side chain. All right, so think of it as a polymer, as a monomer. I told you already. And if it's two monomer, then it becomes a dimer. And such a dimer we will see in the case of glucose. In fact, glucose, like um, sucrose, right? Uh, does anybody know what sucrose is composed of? Sucrose?
Carbon, hydrogen, and oxygen?
No. Sucrose, not glucose, right? Sucrose is a dimer. And what are the two sugar that make up sucrose? Is glucose and fructose. Right? And again, um, sugar usually has the suffix "ose" to it. So sucrose, you see in some fruit, for example, is composed of glucose and galactose. Now, you might have a dimer such as lactose is a dimer of what two sugar? Does anybody know? Glucose and galactose. And there's exists a mannose. Is a mannose is a dimer of glucose and glucose. Okay. So here's an example of a monomer. A monomer and a monomer, if you link them together, the water will come off. This is known as dehydration reaction. Okay. If you break the bond, let's say here dimers to monomer, you would require water. This is known as hydrolysis. You see? Remember we discussed this a couple of weeks ago between what is a synthesis reaction, right? In a synthesis reaction like this is dehydration because water comes off. In hydrolysis or breaks down the molecule, you need water. Okay, hydrolysis. Okay. So also note that, come on, why can't I move this? Okay. So sucrose is a dimer of glucose and fructose. Lactose is dimers of glucose and galactose. And mannose is a dimer of glucose and glucose. Now, some people might drink milk, a lot of milk in, a lot of lactose in milk, and they don't have the enzyme that breaks down glucose to galactose. We'll consider them their lactose intolerance. Lipids, in terms of the lipid dietary fat, is triglyceride. What are the monomers used to make triglyceride again? Does anybody know? What are the monomers used to make triglyceride is the question. So
Glycerin?
make sure you know. What is it?
Glycerin?
Glycerol and 3 fatty acid. Okay, so it's not just glycerol, but triglyceride is 1 glycerol and 3 fatty acid. But other kinds of lipid that you often see is phospholipid. It has a polar and a nonpolar. Molecule that has a polar and nonpolar is known as what? I think of, you know what an amphibian is? Like a frog? Amphi, the word amphi means in between. So amphibians are animals that live in waters and land, right? So amphipathic molecule has both polar and nonpolar. So that's what phospholipid is. Steroids are like estradiols, progesterone, testosterone. It usually ends in "-one". All the steroids in our body has the same structure as cholesterol. In fact, you need cholesterol to make aldosterones, for example, testosterone. Eicosanoid, arcoisanoid are 20 carbon chains that forms a 5 ring. Okay, these are usually small acting molecule in cell signaling. So here's your 4 basic class of lipids, but the most important one that you should actually 2 of them for 301 will be triglyceride and phospholipid. Steroids and eicosanoid, we're going to study more of that in 302, but they're considered as lipid. Okay, triglyceride could have the three fatty acid, right? So here you can say glycerol, sorry, triglyceride is glycerol and three fatty acid. But if I say, oh, triglyceride is broken down to diglyceride. So what is diglyceride will be composed of? What are the monomers that make diglyceride?
Glycerol and two fatty acids?
Good. And monoglyceride?
One fatty acid and glycerol.
Yes, exact. And one glycerol. All right. Now, what's the difference between saturated and unsaturated fat? Unsaturated fat will have, one, double bond. Polyunsaturated, so it will be like omega 5,3, right? Something like that. So if it's more than two double bond, it will be polyunsaturated, which can go through hydrogenation to become saturated, but that's a different story. Remember, saturated fat tends to be solid, like lard. Unsaturated fat, it would be considered oil, which is good for you, not so solid. In adipose tissue, the process of making fat is known as lipogenesis. Whereas if you need a carbon source, where do you break it down from fat? This is known as lipolysis. All right, here is a structural formula that shows you what is triglyceride. Keep in mind that, again, for exam purpose, what is triglycerides made out of? One glycerol, right? All, see that all is an OH, okay, hydroxyl group, we call that. That's an alcohol family, hydroxyl group. Here's the carboxylic group, which is an acid. So this is considered as fatty acid, fatty acid, fatty acid. What makes this acid different from this fatty acid? Because the presence of a double bond. You see here? Phospholipid, we're going to see, is very important in the plasma membrane. It's considered as an amphipathic molecule because it has the polar head, which is hydrophilic, and the fatty acid part is hydrophobic. And typically, draw it looks something like this, where the circle here head is the phospho group, and the tail here would be the lipid. Okay. Now, if it's just single layers like this, there's potential that this could interact with water, but this here lipid right here doesn't like to interact with water. So oftentimes what you see, what the cell did is that it will make two layers of phospholipid. So it'll be like this. Okay. So then anything that is inside of this cell, inside this bilipid two layer, would be considered as lipid. Whereas this right here, the phospho, could be faces the extracellular, and this is considered intracellular, meaning inside the cell. So inside the cells, there's also a lot of water. So basically, the phosphohead will be on the inner layer and the outer layer, if you will. Okay. All right. Steroids. All the steroids come from cholesterol base. An example of steroids are testosterone, estradiol, progesterone. Right. And because some of the destruction of the red blood cells has a structure that called porphyrin ring, which is hemoglobin, and hemoglobin is converted to bilirubin, which is actually part of a bile salt. Okay, that's also steroid, kind of like steroid. Eicosanoid is our 20 carbon ring that has it has a 20 carbon fatty acid, but it has a 5 carbon ring with that as well. And the four classes of eicosanoids are prostaglandin, prostacyclin, thromboxaxins, and leukotriene. all very important in inflammatory response. Now, I say it, I'm not going to ask you about it. I think just focus on the triglyceride and phospholipid. And I guess I told you already the difference between saturated versus unsaturated. Saturated is solid, unsaturated is liquid at room temperature, and fat can be converted to saturation, called hydrogenation. Um, and that's the reason why is they convert unsaturated, to saturated, so that it's in some cases just for manufacturer easy packaging solid stuff as opposed to um, liquid stuff. Peanut oil, for example, to make it peanut butter, they convert it to a hydrogenation process, a saturation process. So partial hydrogenation might lead to trans fat, and trans fats are really bad is because they increase this risk for heart attack. All right, now let's go to the other group of macromolecules known as carbohydrate. Okay, carbohydrate has this chemical formula CH2O, and it's repeated many times. Okay, so it's 6. So here will be C6 times that by 2H12O6. That's the general one I expect you to know in terms of glucose. If it's simple sucrose, glucose, we call it monosaccharide. If it's 2, it's called disaccharide, and it's many, it's polysaccharide. And the question is, what are the polysaccharides that we found in our body? Is known as what is the polysaccharides that you find in our body? Does anybody know? Some people left already. What is it?
Is it glucose?
Glucose is a monosaccharide, one sugar. 2 sugars, disaccharides, will be sucrose. And I expect you to know what is sucrose made out of? Glucose and fructose. Disaccharides, one more time, lactose is composed of glucose and galactose. What's the difference between fructose and galactose, or compound that have same molecular formula but structurally are different, is known as isomer. It's known as isomer. An example of a polysaccharide that you see in our body is glycogen. Yes, thank you, Brandon. I think you answered that last time as well. And the 2 area that we store a lot of glycogen is skeletal muscles and liver. All right, so glucose, 6-carbon monosaccharide, and glucose is the only sugars that we have a lot of enzyme that help breaks down, right? We don't have a lot of enzyme that breaks down fructose completely, for example, but we can certainly break glucose down completely to CO2. But if you ingest fructose, what you have to do is you have to convert fructose to glucose first, right? And in chapter 3, you say glucose and fructose have the same structure, same molecular formula, but different structure, right? They're called isomer. And you can actually, whoever's doing that, thank you, but we really don't need you to do that. And do you know the enzyme that helps convert fructose to glucose is? Okay. It's called isomerase because they're converted to the other. All right, glycogen, you can see here is the polysaccharides that you see in our body. We don't have a polysaccharide known as starch. Plants make starch. We ingest starch, and starch can break down to glucose. The process of breaking down glycogen is known as glycogen lysis. If you make glycogen, it's known as glycogen genesis. Okay, and liver does a lot of that. Here just shows you that glucose is the monomer to make a polymer. You see here, to glycogen, the process is glycogen genesis. If you break it down, it's called glycogen lysis. Okay. Because the monosaccharide, like glucose, has 6 sugar, we call that it's a hexo. Okay. Some sugars only have 5 carbon, in which case we call that a pentose. Okay. So a hexo would be a molecule that has 6 carbon, whereas a pentose, like you see here, will have only 5 carbon. An example of a pentose is ribose, which is a component of the nucleotide we will see. And your job is to find out what are the 3 components of every nucleotide. Well, one of the components is ribose. And the 2 polysaccharide, the 2 nucleic acid molecule would be DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. The difference is between these sugar. One is ribose, the other one is deoxyribose. Okay. Now, because glucose has the same molecular formula as galactose and fructose, but they look different, they're known as isomer. Okay, so isomer, pentose, a disaccharide with 2 sugar, sucrose, is composed of glucose and fructose. Lactose is composed of glucose, galactose, and maltose composed of glucose and glucose. Okay. Okay. Glycogen is a polysaccharides. In plant, they also have cellulose, which is part of their cell wall, plant cell wall, but we lack the enzyme to break down cellulose. Now, if there's a polysaccharide that has a amine group to that, we call that glycosamine glycan. And glycosamine attaches to protein to form proteoglycan. And a lot of our connective tissue, we're going to see later on, as as the matrix in which the cell might sit on, is composed of these proteoglycan. It's just basically a polysaccharide, carbohydrate that has an amine group to that. Okay. And here's an example of the glycosamine glycan, and you can see it's change of glucose. It's called chondroitin, right? Chondroitin sulfate, which might be very important in joint. They play an important role in cell signaling, proliferation, wound repair. This is just basically as an extra, if you don't need to focus on this as much. Here just shows you the difference between a monosaccharide and a disaccharide. Monosaccharides such as glucose, fructose, right, are isomer of each other. See, sucrose is a dimer composed of glucose and fructose. Lactose is a dimer of glucose and galactose. Maltose is a disaccharide of glucose and glucose. Right? Here's another difference between this molecule, this molecule, this molecule, right? They're all 6 carbon. We call that a hexose. But we're here, This is 5 carbon. It's known as maltose. 5 carbons known as pentose, right? Now there's 2 kinds of pentose, deoxyribose and ribose. And this is a typical ribose sugar, which is important of the nucleotide. But what's the difference between ribose and deoxyribose? The word deoxy means without oxy, okay? Particularly at the second carbon. Here's your first carbon, here's your second carbon, third, fourth, fifth carbon, right? The second carbon in ribose has an OH, but this second carbon has an H only, right? So the difference between DNA and RNA, macromolecule is, and the function, what DNA is genetic material. DNA is converted to mRNA, for example. It's the ribose and deoxyribose, and it's all related to this second OH, right? If it's OH is present, it's a ribose. If there's no OH, it's deoxyribose. This is what you see in DNA, and this is what you see in RNA. Right? Okay, so nucleic acid have 2 classes: DNA deoxyribonucleic acid, RNA ribonucleic acid. Okay, and the monomers that makes of these are nucleotides. Okay, so your job is, what are the nucleotides are nucleotide monomer composed of? Every nucleotide, right? In DNA, the nucleotides that makes up are A, G, C, and T. Whereas the nucleotides that make the RNA is A, G, C, and U. Right? Whereas A is, listen to this, is adenosine triphosphate. G is guanidine triphosphate. C is cytidine triphosphate. And T is thymidine triphosphate. Right? And U here is uridine triphosphate. But in short, we just say it's U. And the way that these nucleotide links together is the phosphodiester bond, and an ester is another functional group that has an I think I probably need to show you this. Remember I told you here that this is an acid, right? But if this linked to something else, let's see, an ester. Okay, that's an ester functional group. All right, now the take-home message is this: DNA and RNA is composed of the monomer known as nucleotide. Okay, and what a nucleotide monomer is made out of? It has 3 components: the sugar, the phosphate, and the nitrogenous base. Okay, here's the phosphate group. Here's the sugar. In the case of RNA, that will be an OH. If it's an H here, it will be a DNA. Here's a nitrogenous base. Okay, if it has this structure here, it has 6 ring and 5 over here. It's a purine base. Okay, the base itself has a name. Okay, if the base is adenine, together all of this is adenosine triphosphate, or in this case, it's monophosphate. If the base is guanine, the nucleotide will be guanosine, in this case, monophosphate. If the base is the cytosine, the whole thing, nucleotide will be cytidine monophosphate. If the base is thymine, the whole thing will be thymidine triphosphate. Okay, and so forth. If the base is uracil, the whole thing will be uridine triphosphate, right? So the base has its own name. The nucleotide would have the 3 component: the base, the sugar, and the phosphate has another name. Okay. So there are 3 kinds of nitrogenous base: the pyrimidine base, or your cytosine, uracil, and thymine. Together with the ribose and the phosphate, the cytosine base will become cytidine, right? Monophosphate or triphosphate. And likewise, thymine as a base, but thymine base incorporated into the nucleotide, it will be thymidine, right? The two purine bases are adenine base and guanine base. A base that has a nucleotide that has the adenine base we'll consider as adenosine triphosphate. All right, so here's your nitrogenous base, cytosine, uracil, thymine, adenine, and guanine, right? And remember, uracil as a base, uracils together will form the uridine triphosphate. And uridine triphosphate, for example, is only found in RNA, whereas thymine is the base, the nucleotide is thymidine triphosphate, only found in DNA. All right, so DNA differ from RNA. DNA is double-stranded, and we'll find out that the reason why they're double-stranded because of the pairing of double strand. In one strand, if there's an A, the other strand will be a T. And G is one strand will pair with C on the other strand. Okay. And we'll find out that you can see here thymine paired with adenine, guanine paired with cytosine. So always you call it AT pairing and GC pairing. And then as you move down the along the DNA, you always see the same pairing. Okay? And I wanted to get to our protein, and then this will be included in your exam. The RNA has the base of uracil and no thymine. Okay, typically RNA is single-stranded, and it could be found in the nucleus of cytoplasm, whereas DNA is only found in the nucleus. And we'll talk about the significance of how DNA is used to make RNA in the process of transcription in chapter 3 or chapter 4 and so on. Right? Right now, we just look at the monomer of what are the nucleotides. And again, what are nucleotides is made out of? The three components are the base, nitrogenous base, but you can just say a base, a sugar group, and a phosphate group. Here's a drawing of the difference between DNA and RNA. RNA is single-stranded, you can see here. DNA is double-stranded. Okay? And if you look closely in the RNA, this ribose has an OH here, whereas this ribose here, right here, does not have an OH. That's why it's called deoxy. The DNA is double-stranded like this, and because Watson's and Crick, the two guys who won the Nobel Prize for discover what the DNA structure looked like, we can call one strand is the Watson strand, and the other one is the Crick strand. Okay, it's an anti-parallel position, which means that on one strand there's 5' in, and this refers to the fifth carbon, and this is where the third carbon is. You can add more nucleotide to this, but it's also we always add it at the 3' end. That's not important for you guys at the moment. For students who do research with me, in understanding how you can amplify DNA to the process of DNA polymerase PCR, you have to know the difference between 5' and 3'. What you need to know, and that is, in this case, how A and T pair, and you can have 2 hydrogen bonding, whereas GC has 3 hydrogen bonding. AT is 2 hydrogen bonding. And you can see how hydrogen bonding itself is weak, but is very critical. It allows the DNA to have the double helix structure, if you will. And you will see that hydrogen bonding is weak, but it allows the protein to fold into 3-dimensional structure. One of the most important molecules that we consider as a unit of energy is adenosine triphosphate. It certainly is. It is a nucleotide, but it is a unit for ATP. And you can see here's the base is adenine, here's the sugar, and here's the phosphate. Together, you call this adenosine triphosphate. Okay, and that's your ATP unit. And we essentially is that if you break this bond, okay, uh, this will provide the energy that the cells might be required to do the work. Okay? Uh, to move molecule from an area of low concentration to high concentration, for example, require ATP. And how do you do that? You just break between this phosphate bond, right? So your triphosphate become your diphosphate. And you break the bond further, your diphosphate will become monophosphate. So think of it as this ATP is actually stores the energy in this phosphate bond. And by breaking that phosphate bond, you take the energy that is required for the cells to move molecule, right, from the area of low concentration to high concentration. Okay, lastly, proteins. Proteins are macromolecule, and enzymes is a biological catalyst, but it's also made out of protein. Um, and enzymes will speed up a chemical reaction. We see that. And next time we'll see that how it does that is by lower the activations of energy. But proteins are found abundantly from either cytoskeleton protein, that means inside the cells have formed skeleton. There are contractile proteins like actins and myosin. This plays an important role when we're talking about how muscle contract by actins and myosin slide past each other. It's called the sliding filament model. Protein can make a hemoglobin that you often see in the red blood cell, help carry oxygen. But proteins can have 4 different structure, if you will. It's the same amino acid sequence, but it could acquire different structure. And that's what your job is to find out what are the 4 formation, if you will, that proteins are found in. In the case of hemoglobin, for example, it can find in the form called quaternary structure. And I'm going to talk to you what quaternary structure means, which really mean that it has 2 or more polypeptide. Protein, of course, is made out of amino acids. If it's 20 amino acid or less, we typically call that it's polypeptide. If it's 100 amino acid, then of course it's protein. Okay, but around say if you have 3 to 20 amino acid links together, we call that as a polypeptide. Okay, polypeptide. Poly meaning peptide. Peptide is another bond, a bond between an acid and amino group. It's called polypeptide. If it has 20 amino acid, technically it's protein. Okay, so proteins composed of amino acid. There's 20 different amino acid. Each has, I told you already, an amine and a carboxyl group, and they're linked together. And the amino acid, 20 amino acid, different from each other by this R group. Okay. Amino acid has a N terminus on one end and a C terminus, right? And this one shows you how we call it. This is the N terminus and this is the C terminus. And you can see how the carboxyl group here linked with the amine group, so together is considered as a peptide. So here's one peptide bond, here's another peptide bond, here's another peptide bond. So if it's less than 20, I would call that polypeptide. If it's more than that, like around 20 amino acid, it's considered a protein. Okay. Oligopeptide, small number, okay, or in other words, just peptide. Polypeptides, meaning, but protein is typically is considered as hundreds, if not thousands, of polypeptide. Glycoproteins are protein that have glucose attached to that, which makes it more soluble. I don't expect you to know the 20 different amino acids here, but you can say that you can classify them as either polar, okay, here, and that is the side chains that has a polar. Here's the has non-polar, no charge, but the all amino acid has, sorry, this is your non-polar, this is your polar, and this is your charge, and all of them has amine acid, amine acid, amine acid. Okay. Now let's say that polypeptide bond. Okay, so the fourth structure are primary structure, which is basically polypeptide. Okay, the second structure, due to hydrogen bonding, is this. It's either the alpha helix or the beta plated sheet, and the three-dimensional, that's the secondary structure. The three-dimensional structure called tertiary structure here. Okay, and there are two kinds. They're either fibrous, like this, like collagen fibers, or global into compact, like a lot of enzyme. Okay, tertiary structure. And finally, if it has more than one polypeptide, so in other words, an example of that hemoglobin is called quaternary structure. Two or more polypeptide chains together is considered a quaternary structure, and that's the hemoglobin. Okay, and in the lab that you often will see that enzymes or biological catalysts that speed up a chemical reaction. If you subject the enzyme or degradations of starch by amylase, right, or amylase breakdown of starch, at extreme pH, it will not work because pH will break down the hydrogen bonding. Okay, denature that protein. When protein comes apart, we call that denature. And certainly heat can do that as well. This is the consequence of extreme heat, and that is you can denature some of your protein. All right, so that will be the conclusion of chapter 2. Your exam will be up to this. Most importantly, what are the four macromolecules, what are they make, what are the monomers that makes them, and in terms of protein, please focus on there are four structure, I guess you would say, that polypeptides or protein can be found either as one polypeptide, and two, either the alpha coil beta plated sheet, three, tertiary structure, global or fibrous, and then quaternary structure in which two or more polypeptides form. An example of that would be hemoglobin, where it has two alpha chains, two beta chains, or even antibodies. Okay, our antibody is a quaternary structure because it has two heavy chains and two light chains. All right, any question? If there's no question, I'm going to turn off the recording.